Hello, my dear students. Welcome back to another exciting journey through the wonders of animal biology. Today, we dive deep, literally, into the fascinating topic of biology. Have you ever wondered how a fish survives in salty ocean water, or how a bird that drinks seawater stays hydrated? The secret lies in a remarkable biological process called osmoregulation. Osmoregulation is like the body's internal pumping system. It regulates the water and salt balance to keep everything running smoothly, whether it's preventing the body from turning into a water balloon or drying out like a sponge. Osmoregulation ensures that the internal environment stays just right, but how do animals actually do this? External osmoregulatory organs are special structures that help in this water and salt balance. Gills in fish, not just for breathing. In marine fish, gills help expel excess salt, while in freshwater fish, they absorb needed salts from their surroundings. Salt glands in birds and reptiles are the superglands are nature's desaltination unit found near the eyes, nostrils or tongue. They remove extra salt from the body so efficiently that some seabirds literally drip salt from their beaks. The ultimate goal, what is the ultimate goal? Homeostasis. It is maintaining a stable internal environment no matter how salty, fresh or dry the outside the world is. So whether a goldfish in a bowl or a seagull soaring above the sea, these creatures owe their survival to the incredible teamwork of their osmoregulatory organs, silent heroes working behind the scenes to keep life in balance. Types of osmoregulation. There are two major types of osmoregulatory strategies used by various organisms. First one is osmoconformer. These organisms conform to the osmotic pressure of their surrounding environment. Their internal osmolality is approximately equal to that of surrounding water. They do not actively regulate the concentration of solutes in their body fluids. Mostly found in marine environments where salinity is relatively stable, they are less energy consuming as no active osmoregulation is involved. Their examples are most marine invertebrates like echinoderms, sponge, cnidarians, some marine fishes like hagfish. Osmoregulators, these organisms keep their internal osmolality constant despite external changes. They actively regulate water and salt solute balance through physiological processes. Osmoregulation is often done via organs like kidneys, gills, salt glands or skin. They require more energy due to active transport of ions found in freshwater and marine environments as well as terrestrial habitats, and the examples of osmoregulators include freshwater and marine bony fishes, amphibians, reptiles, birds, mammals, etc. Now we'll study osmoregulation in fish. Fish living in freshwater and seawater regulate both salt and water balance in different ways due to differences in the salinity of their environment. This is the diagram of osmoregulation found in freshwater fish and in marine fish. What happens in case of freshwater fish? Freshwater fish live in hypotonic environments which is less salty than their body fluids. Their body fluids are hypertonic or hyperosmotic, meaning that they have more salt than the surrounding water. Osmoregulatory challenges. Freshwater fish face or exposes to main problems. Water continuously enters their body by means of osmosis, which causes risk of swelling. Salts keep diffusing out of their body into the environment, leading to loss of essential salts. There are adaptations to prevent water gain or salt loss. They do not drink water to avoid more water entering the body. Their kidneys produce large amount of dilute urine to remove excess water. The important salts are reabsorbed from the kidney tubules into the blood. The special gill cells actively absorb salts like Na plus and Cl minus from the water into the blood. Active salt absorption. Gill cells can actively transport salts from surrounding water even when the water has very low salt concentration, that is just greater than 1 millimole per liter. They can move salt against a gradient of over 100 times the external concentration. Role of skin or integument. The skin is less permeable or impermeable to both water and salts. They help to reduce salt loss and water gain. Now, what is the conclusion? This is the diagram of osmoregulation through in freshwater fishes. Freshwater fish prevent excess water gain by making dilute urine. They prevent salt loss by actively absorbing salts through gills and reabsorbing salts in kidneys. Their skin and behavior, not drinking water, also help in osmoregulation. Osmoregulation in marine fishes or seawater fishes. কি বলছে basic osmotic challenge. Marine fishes live in hypertonic environment which is saltier than their body fluids. Their body tends to lose water to the environment, gain salt from the surrounding seawater. General adaptation in marine fishes. They drink large amount of seawater to compensate for water loss. They excrete small amounts of concentrated urine to conserve water, actively expel excess salts using gills which mainly excrete Na+, Cl- and sometimes K+. Kidneys which mainly excrete divalent ions like Mg2+, Ca2+, and sulfate 2+. The rectal glands are found in case of elasmobranchs like sharks and rays. Different categories of marine fishes and their strategies. Hagfishes, although it is not fish, belong to fish, they are called as myxine. Their plasma is osmotic to seawater. They require less energy for osmoregulation. They maintain lower levels of Ca2+, Mg2+, and sulfate 2-. They have higher levels of Na+ and Cl- than the surrounding seawater. The sharks, rays, skates, elasmobranch and coelacanth, latimeria. Their plasma also isosmotic to seawater. They maintain low concentration of inorganic ions. They use organic osmolytes like urea, trimethyl amine oxide, TMAO for osmotic balance. Their excess NaCl is excreted via kidneys and rectal glands. Marine teleost or marine bony fishes, their body fluids are hypotonic to seawater. The water lost occurs through gill epithelium. They can compensate by drinking seawater, absorbing Na+ and KCl- which is approximately 70 to 80% via intestinal lining. They excrete unabsorbed divalent ions like calcium 2+, Mg2+, SO4 2- through kidneys. Excess monovalent ions, they are excreted via gills using active transport. Urine and salt balance. Urine is isosmotic to blood. Urine contains high levels of divalent ions which are not excreted by gills. The gills and kidneys together help in retaining water and eliminating excess salt. Special cases we can found in case of salmon, which is a migratory fish. They live in both freshwater and marine environments. They can adjust osmoregulatory mechanisms to maintain a constant plasma osmolarity during migration. A classic example of euryhaline species, this is salmon, is a classic example of euryhaline species. Classification of fishes based on osmoregulation according to Mole and Czech Jr. 1982. Fishes can be divided into 4 major groups based on how they regulate internal water current and total solute concentration or osmolarity. The first one is the stenohaline osmoregulator, which actively takes strategy and their salinity tolerance is narrow. They osmoregulate and it is found in carp and tuna, etc. Euryhaline osmoregulators, which is also active but the salinity tolerance is very wide. The regulation type is osmoregulation and it is found in salmon, eel, etc. Stenohaline osmoconformer, their strategy is passive in nature and it is salinity tolerance narrow. Their regulation type is osmoconformation and it is found in hagfish. Euryhaline osmoconformer, they are also passive and the wide range of salinity tolerance. Their osmoconformation is the type of regulation there and some elasmobranchs show the phenomena. So marine fishes face constant water loss and salt gain due to their hypertonic environment. They drink seawater and excrete excess salts through gills, kidneys and rectal glands. Hagfish are isosmotic while elasmobranchs and teleost actively regulate ions differently. Some fishes like salmon maintain stable plasma osmolarity even while migrating between freshwater and marine environments. Salt glands in reptiles. Salt glands help reptiles remove excess salt, especially when their kidneys can't produce urine more concentrated than blood. These glands are found in marine iguana and lizards, sea snakes like Laticoda or semifasciata, marine turtles, crocodiles and other terrestrial reptiles. Salt glands are often located in the tongue. Their function, they help secrete a highly concentrated sodium chloride solution. It's an adaptation to environments with high salt exposure like the ocean. In sea snake or Laticoda semispeciata, they excrete dilute urine from their kidney, which is 7 to 148 millimole Na plus. They secrete concentrated salt 380 to 625 millimole sodium NaCl through their mouth via salt glands. Salt excretion increases after salt or sucrose intake, proving active salt gland function. Salt gland location by species: lizards, nasal glands. Salt exits through the nostril. Turtles, hadrian glands are near the eyes. Their salt exits under the eyelids. Crocodiles likely excrete salt via glands in the tongue and possibly near the eyes. Conclusion: salt glands provide extra renal outside kidney help for electrolyte balance. This is especially vital for marine and desert reptiles with limited fresh water. Now we will study salt glands in birds. Salt glands help marine and desert birds maintain salt balance without drinking fresh water. Birds with salt glands include marine birds like cormorants, cormorant gulls, terrestrial birds like ostriches. Here the images of cormorant, gulls and ostriches. Their function to excrete or secrete salt NaCl solution, then concentrated hypertonic NaCl solution they secrete. These glands are especially important when birds consume salty food or sea water. The gland has many small lobes about 1 millimeter in diameter. Each lobe has secretory tubules that drain into a central canal. The canal connects to a duct running through the beak and opens at the nostril. Their countercurrent mechanisms: blood in capillaries flow opposite to the direction of the secreted fluid. This helps in efficient concentration of salt in the glandular fluid. Parasympathetic nerves regulate the salt gland activity in birds. The corticosterone and arginine in vasotocin also stimulate salt secretion. So, external osmoregulatory organs in vertebrates play a crucial role in maintaining the balance of water and electrolytes by actively regulating salt and water exchange with the environment. These specialized structures, such as salt glands in marine birds and gills in fish, enable vertebrates to survive and thrive in diverse aquatic and terrestrial habitats. Thank you for watching today's class. If you found this video helpful, don't forget to like, share and subscribe to the channel for more student friendly biology content. Until next time, stay curious and keep learning. See you in the next class. Thank you.